Assalamu alaikum dear students. Today the topic that we are going to discuss is factors affecting erythropoiesis. The four factors that you have to remember are iron, vitamin B12, folic acid, few nutrients and erythropoietin. These all are very important for the process of erythropoiesis to take place. First of all, let's start with the iron. As we all know that RBCs have a prime component known as hemoglobin, and hemoglobin comprises of the heme part which has iron in it. So iron is very important for the production of hemoglobin. We get iron from different sources, like from fruits comprising of apple, pomegranate, then a few vegetables that are spinach and different kind of vegetables that have green leaves to them. Now whenever iron is ingested, it enters into the iron enters in the form of Fe3+, that is ferric form, through the food into the stomach, and from there it enters into the duodenum. Now in the duodenum the iron is absorbed, and how it is done, let us discuss it in detail. First of all, at the luminal end there is an enzyme which will cause the conversion of ferric form of iron, that is Fe3+, to be converted to the ferrous form, that is Fe2+. Now this Fe2+ will enter into the duodenal cells through a channel. In the duodenum we have a protein which is known as apoferritin. This protein is known as apoferritin. Now whenever the iron molecule gets attached to this apoferritin, this new structure is then known as ferritin. So the protein that is without the iron attached to it is known as apoferritin, and when the iron attaches to it, it is known as ferritin, and ferritin is basically the stored form of iron. Now there is cluster of this ferritin present in the cell whenever the ferritin levels exceed. This cluster of ferritin or polymers of ferritin is known as hemosiderin. Now let's discuss that how this iron will enter into the bloodstream. As ferritin is the stored form of iron, this ferritin gets digested by the lysosomal action. Lysosomes cause the release of iron from the apoferritin, and hence this iron will then move through these channels at the basilar end of the duodenal cell out of the cell into the interstitium, and this ferrous ion will then be converted by the enzyme into the ferric ion. Now after the conversion of ferrous to ferric form, let's make it a bit clear. Now this ferric will enter into the bloodstream, and in the blood we have a special kind of plasma protein which is known as apotransferrin. To this apotransferrin, the iron gets attached to it and forms another structure which is now known as transferrin. So transferrin is basically the transport form of iron. That is, iron gets transported from the GIT to the rest of the body in the form of transferrin through the bloodstream. It may enter into the liver for the storage purpose. It may enter into the muscle for myoglobin formation. It may enter into the bones for the formation of the RBCs. So from there, in the form of transferrin, this iron will then approach the vessels present in the red bone marrow. Now that was it for the iron. Now let's talk about the B12 and folic acid. Again, we get these components, that is folic acid and B12, from the food that is ingested. Now let's talk first about the suppose we have these two substances. One is B12. This green one is showing B12, and then we have this, I should change the color to some good old one. Okay, and then we have this blue one which is known as folic acid. Over here I'll just write it with Fa. Now let's first see what happens to the folic acid. The folic acid moves from the stomach into the duodenum, and at this level it is engulfed through endocytosis into the cell, the duodenal cells, and from there it is exocytosed out into the bloodstream. Fine. Now the most important one, the important one, and the one that you have to memorize is the B12. B12 is a vitamin that gets absorbed at the level of the ileum. Now in order of B12 to move from stomach into the ileum, it gets digested. So the stomach is specialized to have the production of intrinsic factor. So the stomach produces a factor which is known as the intrinsic factor. We all have, you guys need to remember this thing, that the parietal cells present in the stomach produce the intrinsic factor. Now what happens, that this intrinsic factors combines with the B12 molecule. It combines with the B12 molecule, and this complex then cannot be digested in the stomach, in the duodenum, in the jejunum, or at the level of ileum. So this structure, that is the intrinsic factor, along with the B12, will move through the intestine and will reach to the ileum. At the level of ileum, The ileal cells have a receptor present at their luminal end to which the intrinsic factor gets attached. So to this receptor the intrinsic factor will get attached, and as we all know, this intrinsic factor is attached to the B12 ion. So through the process of endocytosis, receptor mediated endocytosis, a vesicle will be formed which will be containing the B12 molecule along with the intrinsic factor in it. So this vesicle will then be exocytosed and there will be release of the B12 over there in the blood. So now we can see iron released into the bloodstream, we can see folic acid released into the bloodstream, and we can see B12 released into the bloodstream. Now the fourth and the primemost and a very, very, very important hormone, that is the erythropoietin. Let's discuss it in detail. As we all know, erythropoietin is released from the kidneys. In the kidneys we have these nephrons, and it is not very clear till yet that from which level of the nephron the erythropoietin is released, but still there are a few studies that say the proximal convoluted tubule cells have the intrinsic activity of production of erythropoietin in them. So what happens that whenever there is hypoxia, before hypoxia, let's see that this is the cell of the proximal tubule which has a nucleus to it. Along with it there is a DNA, and this red part of this DNA is the one which is the erythropoietin gene. This gene has the code for erythropoietin formation. Now what happens is that we have a structure over here, and this transcription element, basically this transcription factor, is known as hypoxia inducible factor 1. So hypoxia inducible factor 1, this is the factor which, when combines to this erythropoietin response element present on the erythropoietin gene, causes the production of erythropoietin. But as we know, we do not need erythropoietin all the time. So this factor is basically controlled by an enzyme which holds to it. There is an enzyme which holds to this hypoxia inducible factor 1 through hydroxylation. So whenever there is hypoxia, this hydroxylation will not take place. And the grip, or you can say the hold of this enzyme to the hypoxia inducible factor 1 will be released. So this hypoxia inducible factor 1 will go to the nucleus and will get attached to the erythropoietin response element, leading to the formation of protein which is known as erythropoietin. And erythropoietin is, as I have told you guys before, it is very, very, very important for the production of RBCs. Now this erythropoietin will then get into the bloodstream as well. So now you can see how iron has entered into the bloodstream, how B12 has entered into the bloodstream, and how erythropoietin has entered into the bloodstream. Similarly, carbohydrates, amino acids and fats get absorbed through the GIT, and they also enter into the bloodstream. Now this whole blood, when reaches to the bone, now let's see what will happen inside the